Unleashing the embryo's potential, embryo development is a fascinating and complex process that marks the beginning of life. It starts with the union of a sperm and an egg, leading to the formation of a single cell called a zygote. This initial stage sets the foundation for all future growth and specialization. Fertilization occurs when a sperm successfully penetrates an egg, combining their genetic material. This union results in the creation of a zygote, a single cell with a complete set of chromosomes. This crucial event marks the start of embryonic development and initiates a cascade of cellular processes. The role of the sperm and egg is pivotal in the development of an embryo. The sperm provides half of the genetic material and triggers the egg's activation. The egg, on the other hand, supplies not only the other half of the genetic blueprint, but also the nutrients and environment necessary for early development. Once fertilization occurs, the zygote begins its journey of rapid cell division known as cleavage. Through a series of mitotic divisions, the single-celled zygote divides into multiple smaller cells. These early divisions are critical for increasing the cell number without increasing the overall size of the embryo. As cleavage progresses, the embryo reaches the morula stage, characterized by a solid ball of cells. The cells, called blastomers, continue to divide and compact, preparing for the next stage of development. The morula represents a key step in the transition from a simple cluster of cells to a more complex structure. During the morula stage, the embryo resembles a solid ball of cells. This stage is crucial as the cells, called blastomeres, begin to differentiate. The morula, typically formed about 4 days after fertilization, prepares to transition into the next stage, setting the foundation for future development and implantation. Following the morula stage, the embryo transforms into a blastocyst. This structure contains an inner cell mass and an outer cell layer called the trophoblast. The blastocyst's formation is essential for implantation and the eventual development of the embryo's various tissues and organs. Implantation occurs when the blastocyst attaches to the uterine wall. This process is vital for establishing a connection between the embryo and the mother. Successful implantation ensures the embryo receives necessary nutrients and signals, marking the beginning of a symbiotic relationship crucial for continued development. The trophoblast, part of the blastocyst, plays a significant role in implantation. It invades the uterine lining, facilitating nutrient exchange and forming the early placenta. The trophoblast development is critical for establishing a supportive environment, ensuring the embryo's growth and the pregnancy's progression. An embryonic disc forms from the inner cell mass of the blastocyst. This flat, circular structure will eventually give rise to the embryo itself. The embryonic disc's formation marks a pivotal point in development, as it sets the stage for the differentiation of the primary germ layers. Gastrulation marks a critical phase in embryonic development, where the single-layered blastula reorganizes into a multi-layered structure. This process establishes the 3 primary germ layers: Ectoderm, Mesoderm, and Endoderm. Each layer will give rise to specific tissues and organs, setting the foundation for the embryo's complex anatomy. The ectoderm is the outermost germ layer formed during gastrulation. It differentiates into the nervous system, including the brain and spinal cord, as well as the skin, hair, and nails. This layer also contributes to the development of sensory organs, such as the eyes and ears. The mesoderm forms between the ectoderm and endoderm and is responsible for creating the body's structural components. It gives rise to muscles, bones, and the circulatory system, including the heart and blood vessels. The mesoderm also develops into the kidneys and reproductive organs. The endoderm, the innermost germ layer, lines the developing gut and respiratory tracts. It differentiates into the gastrointestinal tract, liver, pancreas, and lungs. This layer is essential for forming the body's internal organs and plays a crucial role in nutrient absorption and gas exchange. The formation of the primitive streak is a key event in establishing the body's bilateral symmetry. This structure appears along the midline of the embryo and serves as a guide for cell movement during gastrulation. It defines the embryo's head-to-tail axis and initiates the formation of the germ layers. Neurulation marks the pivotal stage where the neural tube forms, eventually developing into the brain and spinal cord. This complex process involves the folding and closure of the neural plate, ensuring the foundational structures for the central nervous system are established. proper neurulation is critical for healthy neurological development. Somate formation follows, segmenting the embryo into repeating units. These somites eventually differentiate into essential structures such as vertebrae, ribs, and skeletal muscles. Their orderly formation is crucial for the organized development of the body's axial skeleton and associated musculature. The notochord forms along the embryo's midline, serving as a primitive backbone. This rod-like structure provides essential signals that guide the development of surrounding tissues. The notochord's role is crucial in establishing the embryo's axis and inducing the formation of the neural tube. Heart tube formation is a critical milestone, marking the beginning of the cardiovascular system's development. This tube will loop and fold to form the primitive heart, which starts beating and circulating blood. Early heart development is vital for ensuring the embryo receives necessary nutrients and oxygen. Limb bud formation signifies the initial steps in developing arms and legs. These small protrusions emerge from the embryo's sides and will eventually differentiate into complex limb structures. Proper growth and patterning of limb buds are essential for the formation of functional extremities. Organogenesis marks the critical phase where the embryo's organs begin to form. During this period, cells differentiate and assemble into complex structures, laying the foundation for functional systems. This intricate process ensures that each organ develops with precision, preparing the embryo for survival and growth. The placenta's development is vital for sustaining the growing embryo. It facilitates nutrient and oxygen exchange between the mother and the embryo while removing waste. This temporary organ also produces hormones that support pregnancy and fetal development, ensuring a stable and nurturing environment. The circulatory system begins to take shape early in development, with the heart forming as a tube. Blood vessels branch out, connecting the embryo and placenta. This system ensures efficient nutrient and oxygen delivery, enabling rapid growth and the proper functioning of developing organs. The respiratory system's development starts with the formation of lung buds. These buds branch into intricate structures that will become the lungs. Although the fetus doesn't breathe air, the respiratory system prepares for post-birth breathing, ensuring the newborn can efficiently exchange gases. Digestive system development involves the formation of the gut tube, which will differentiate into the stomach, intestines, and other digestive organs. This system prepares the fetus to digest and absorb nutrients after birth, ensuring its ability to grow and thrive independently. The development of the urinary system begins with the formation of the pronephros, mesonephros, and finally, the metanephros, which becomes the permanent kidney. As the embryo grows, the kidneys ascend to their final position, establishing connections with the bladder and urethra for waste elimination. The reproductive system's development involves the differentiation of the gonads into either tests or ovaries, influenced by genetic and hormonal factors. Internal and external genital structures form, ensuring the future capability for reproduction. This process sets the foundation for male or female reproductive anatomy. The brain and spinal cord originate from the neural tube, which initially forms as a flat sheet of cells and then folds. Various regions of the brain develop, each specializing in different functions. Neurons proliferate and migrate, establishing the complex network necessary for central nervous system activities. Eye and ear development are crucial for sensory perception. The eyes form from the optic vesicles, which give rise to the retina, lens, and other structures. Similarly, the ears originate from the otic placodes, evolving into the inner ear's intricate components responsible for hearing and balance. The skeleton begins as a cartilaginous framework, which gradually ossifies to form bones. This process, called endochondral ossification, involves the replacement of cartilage with bone tissue. The axial skeleton, including the spine and skull, forms first, followed by the appendicular skeleton, which comprises the limbs. Muscle development in the embryo begins with the formation of somites, which are segmented blocks of mesoderm. These somites differentiate into myotomies, giving rise to skeletal muscles. As the embryo grows, muscle fibers lengthen and thicken, eventually forming the muscles that will control movement and support the body. Skin and hair formation start with the outermost germ layer, the ectoderm. The skin begins to form in layers, providing a protective barrier for the developing embryo. Hair follicles also emerge from the ectoderm, setting the stage for hair growth that will continue after birth. The immune system begins to develop early, preparing the embryo to fight infections. The thymus and bone marrow, essential for immune function, start to form. White blood cells are produced, which will play a crucial role in identifying and neutralizing pathogens, ensuring the embryo's future health. The endocrine system's development is key for regulating numerous bodily functions. Glands such as the thyroid, adrenal, and pancreas start forming, releasing hormones that control growth, metabolism, and stress responses. These hormones are vital for the embryo's development and will continue to regulate bodily functions throughout life. Fetal movements and reflexes become noticeable as the nervous system matures. Spontaneous movements can be observed, indicating muscle and nerve development. Reflexes, such as sucking and grasping, also appear, preparing the fetus for survival outside the womb. These movements are crucial for muscle strengthening and coordination. As the fetus grows and matures, it undergoes significant physical and functional changes. Organs develop and refine their functions, while bones harden and muscles strengthen. The fetus accumulates body fat, vital for temperature regulation after birth. These changes prepare the fetus for the demands of life outside the womb. Fetal viability refers to the fetus's ability to survive outside the uterus, typically around 24 weeks of gestation. Advances in medical technology have improved survival rates for preterm infants. However, the chances of survival and potential complications vary, emphasizing the importance of prenatal care and monitoring. Genetic factors play a crucial role in fetal development, influencing traits and susceptibility to diseases. Environmental factors, such as nutrition, exposure to toxins, and maternal health, can impact the fetus's growth and development. Understanding these influences helps in promoting a healthy pregnancy and reducing risks. Developmental disorders can arise from genetic mutations, environmental exposures, or complications during pregnancy. Common disorders include neural tube defects, congenital heart defects, and Down syndrome. Early detection through prenatal screening and diagnostic tests allows for timely interventions and support for affected families. The journey from conception to birth is a complex, awe-inspiring process. Each stage of development is crucial in forming a healthy human being. Advances in science and medicine continue to enhance our understanding of this miracle, offering hope and better outcomes for future generations. As the fetus continues to grow, it undergoes significant changes in size and complexity. The organs and systems that formed earlier start to mature and become fully functional. This phase is crucial for ensuring the baby can survive outside the womb, with key developments in the brain, lungs, and immune system. External factors like maternal health, nutrition, and environmental influences play a vital role in fetal development. These factors can affect everything from growth rate to organ function. Understanding these influences helps in promoting healthy pregnancies and minimizing risks. Ultimately, the journey from embryo to birth is a testament to the resilience and complexity of life.